All right. Hi. Yeah, so I'm Jay Greenbaum, and I'm excited to be able to tell you about the um, T-cell class 1 tools, the binding, processing, and immunogenicity tools, and kind of the integrated app that we've developed on the next-gen tool site. So we're going to start with an overview of the endogenous antigen processing pathway for class 1. And we're going to use this cartoon kind of throughout the presentation to kind of keep ourselves oriented uh, with the different parts of the workflow, parts of the process that we're talking about. What this cartoon is showing is that in all cells, proteins are constantly being broken down by the proteasome into shorter peptides. Some of those short peptides get transported into the ER by the TAP protein. Some of those peptides will bind to specific MHC molecules. And then those MHC peptide complexes will make their way to the cell surface where they may or may not be recognized by a CD8 positive T cell. And we could break down the different parts of the pathway into the processing portion. So this is where the portion of the pathway that the processing tools predict. This is the breakdown of proteins into peptides and transport into the ER. Then the MHC binding portion. And finally, the immunogenicity portion of the pathway. And we can start with kind of a description of the different data types that we use to train our models to make predictions for these parts of the pathway. And we start with MHC binding, which is an in vitro assay that measures the interaction strength of a specific peptide MHC complex. So we get in vitro binding strength data, kind of one data point per assay. The next group of experimental data types are MHC ligand dilution data, where we have APCs with known MHC types, and we can elute peptides from the cell surface and get generally thousands of data points, thousands of peptides that we know can bind to one or more of the MHCs. And an interesting product of that data type is that there we only get positive data. So we only get a list of peptides that we know bind. We don't get any negative data. And then finally, there's T cell response assay data, where we can measure cytokine release from T cells that have bound, whose TCR have bound a MHC at the cell surface of an APC and release cytokines. And that's where we use, that's the type of data that we can use to train immunogenicity models. And if we talk for a moment about the class 1 MHC molecule, this molecule is expressed by almost all nucleated cells in vertebrates. And it's a 2-chain molecule with a short constant chain called B2M or beta-2 microglobulin, and then a longer variable chain that's very highly polymorphic with thousands of known alleles. And this is the only chain that actually impacts binding. So as you can see on the right, it's the alpha chain that forms the binding cleft for peptides. And that binding cleft is relatively short and relatively closed so that it can accommodate only short peptides of length 8 to 11. So to go from data sets to prediction tools, generally all prediction tools work in the same general way where we start with a training data set. And here we have a binding data set as an example where we have known MHC types, we have known sequences, and we have known measured binding affinities. And we can throw that at various prediction models, neural networks, linear weighted models to develop a trained model. And then we can take that trained model with unknown data. So here we have just peptides and alleles, and then hopefully get a close prediction to what the actual binding affinity. So we go from input data to the output data where we have the binding score predicted. So most of the models are more or less follow that kind of training. And now if we back up to MHC binding prediction, which again is this part of the pathway, and on our NextGen tool site, you would get there by going to this URL, nextgentools.iedb.org/tc1. What I'm showing here is actually the homepage of the site. So the TC1 link can be accessed through here or by hitting this play button here. If you go from the play button, you'll end up populating the form with example data, and you'll have a prediction running once you get to the next page. So this is what you'll find on the next page. This is now a big input sequence box. This is where you would put your antigen sequence or list of peptides that you would like to make predictions for. And to get your data into that box, you could type it in, you can copy and paste it in, or you can just drag a file over. And once you do, the contents of that file will be populated in that box. So the next step would be to select the other parameters that are below the input sequence box. So we can go to our peptide lengths and our MHC alleles. And in terms of alleles, there's a way for you to enter alleles in one by one. So we have a nice autocomplete feature that will allow you to just start typing an allele name and you'll get matching alleles. You could just select them one by one, or you could use one of the panels that we have available through the allele finder. And so, for instance, we have this reference set of 27 alleles that covers 97% of the population, meaning that if you have a pool of peptides where you have at least one peptide in that pool that binds to at least one of these alleles, theoretically, that pool of peptides would cover about 97% of the global population. So to use the reference panel, you would just click on the allele finder button, which would bring up this pop-up window. And you can then click on the 27 allele panel, which will fill in, and then click submit. And then that will populate your allele selection box with the list of 27 alleles. In terms of lengths for selection, what we're showing here on the left-hand side is the relative binding affinity of 5 different alleles over various peptide lengths. And what we can see is that the kind of preferred profiles for binding for various alleles kind of varies with it's kind of not consistent between the different alleles. So for instance, the HLA A0101 profile is able to bind longer peptides in general, whereas the HLA-B 5101 peptide kind of peaks at a length of 8. In contrast, on the right, we have peptides that are actually eluded from the cell surface for these same 5 alleles. And here we can see that there's a very sharp peak at length 9. And also, many of them will elute peptides of length 10. And so we just recommend taking this type of information into account when you're making your length selection, thinking about the goal of your analysis. All right, so we've entered our sequence, we've made our peptide length selection, we've selected our alleles. Now we can select the prediction model that we want to apply to the data. And here we are selecting the NMHC PAN 4.1 BA model. And we do have several recommended methods. And we currently have a recommended epitope predictor, which is NMHC PAN 4.1 EL, and a recommended binding predictor, which is NMHC PAN 4.1 BA. The difference being that EL is trained on elution data, so peptides that are known to be eluted from the cell surface and have gone through most of that processing pathway. Whereas the BA predictor is trained on in vitro binding data and predicts simply the part of the pathway that is binding to MHC. And our recommendations have kind of changed over the years, and they'll continue to change. And that's based on what we observe from benchmark data. And on the right-hand side, we're just relaying the network architecture of an MHC PAN 4.1, which is a neural network predictor that takes as input a peptide and an MHC pseudosequence and will simultaneously output a binding affinity value and an eluted ligand score. In terms of the benchmarks, so we do host this page on the Classic site where we, every week, we check for any new data deposited into the IEDB that is MHC binding data. And we can run that data through this whole list of predictors and determine how well do each of the predictors kind of recapitulate the measured binding affinities. And we've been doing this for quite a while, and we have this kind of rolling metric of prediction scores. And so we can use this to inform what we consider to be our recommended methods. So now we've entered all the parameters. The last thing to do is to click run. And then once we do that, the sequence will get broken up into peptides of all of the lengths that we selected. And then the binding affinity or elution score for each of the peptide MHC combinations will be predicted. That binding score will then be compared to that of a large set of randomly selected peptides so that we can assign a percentile rank. And then if we do select multiple methods, we'll get a median percentile rank calculation as well. And so just a word on percentile ranks and why we calculate them. Here we're showing the predicted IC50 values for the HLA-A0201 allele. So this is kind of what the distribution looks like from high affinity, low percentile rank, low IC50. So a low IC50 is high affinity to low affinity. And if we compare that to a different allele, so HLA-A2402, we can see that this distribution is shifted way over to the right. And then zooming in to our recommended cutoff of 1 percentile, we can see that if we simply used a hard IC50 value cutoff for the two different alleles, we would end up with very different results with different numbers of peptides per allele. So we can see, for example, the 1 percentile for HLA-A0201 hits at about 27, and the 1 percentile for HLA-A2402 is at 511. And so the percentile rank is just a nice way to normalize the values and control for the allele-specific or predictor-specific effects. All right, so while our prediction runs, we'll get a spinner that looks like this, depending on the size of your data set, that can last from several seconds to minutes to even potentially hours. And eventually, you'll end up with a table that looks something like this, where you'll have kind of one row per peptide and allele, and then the various columns corresponding to that pair. And it will by default be sorted from low to high percentile rank. So by default, we have a set of columns that we think are the most meaningful, but we do make other columns available to you. And you can get at them by clicking on the Display Columns button, which will bring up this modal where you can select or deselect the columns that are most relevant for your analysis. And you can also get more information on each of the individual columns from here. One other functionality that we have is to be able to download the prediction results. So you could download either the full data set or just the rows that are displayed, meaning that if you've applied filters, you can just limit your download to that data set. And we provide the option for several different formats, so TSV, CSV, or JSON. And once you download, we'll get a TSV file or CSV file that looks something like this. And you can take that and you plug into your own workflows. One more functionality that we have specifically for longer running predictions is to provide a pipeline name and email address so that when you do that, you will get an email with a link that goes back to the pipeline. And when the prediction is done, you'll be able to get back to the pipeline in its state, see all the inputs and outputs, and you can even share that link. All right, so we've done our prediction. We have this big table of predicted values. What do we do with it? How do we select our binders? And our recommendation is that we don't have a recommendation. We have several kind of preferred approaches that we've used over the years in various studies, and they can all be sane and rationalized for your particular use case. And so just going from top to bottom, one possibility is to pick all of the peptides that are below percentile rank of 1, which should capture many of the actual binders. Another option is to, instead of taking the percentile rank less than 1, just pick the top 1% of peptides overall for each allele and length combination. And that'll give you an equal number of peptides per allele. You can also use a hard cutoff, such as 500 nanomolar, which has kind of been the classically defined intermediate binding affinity for MHC class 1. And by selecting a hard cutoff like that, that will ensure that all of the peptides have a reasonable affinity. And then we have developed some allele-specific binding affinity thresholds as well. And you can find more about those by going through the help, where we have a table of the various alleles and their allele-specific cutoffs. And that comes from this paper that's linked here. So all of the approaches are reasonable and have been applied in numerous studies. You can even combine the thresholds. For instance, take the peptides that are in the top 1% and that are also below 500 nanomolar. And past studies suggest that allele-specific thresholds may be useful as well. So moving on from class 1, now we look at the processing tools, which is this part of the pathway. The moving from the full-length peptides through the proteasome into the ER. And we get at the tool through the same link. And when we select now processing instead of binding, we get presented with a few options. We have several processing methods available. Here we're going to talk about the basic processing prediction. And this is a predictor that combines predictions for MHC binding, proteasomal cleavage, and tap transport. So that part of the pathway that we highlighted. And you can see when you select this method, there are a few context-specific parameters that come up. And we generally recommend just kind of leaving these parameters at their default, but we do leave them there for you to fiddle with if you have specific needs and if you're familiar with what they do. So we have parameters related to proteasome cleavage on the left that are related to the sequence motif that's used to predict cleavage, and then parameters related to tap transport on the right-hand side that relate, excuse me, that relate to how far downstream in the sequence to look for the signal. So once we select our parameters, again, we just click run, and we'll end up with a table that will look similar to the output for our binding and elution tools, but now instead of binding, predicted binding affinities, we're going to have predictions based on the processing. So we'll have a column for proteasome score, for TAP score, MHC score, and with this tool, for all of the score columns, a higher score is better. And we've transformed the MHC binding affinities to, we've done a negative log 10 transform to those affinities that changes the sign and also changes it such that higher is better. And then we have our summary scores, which is this processing score is simply the addition of the sum of the proteasome and the TAP score. And then the processing total score adds onto that the MHC score. And so we recommend using this tool as a companion to the binding tools to kind of help understand why a peptide might not be a good ligand despite strong predicted binding. Most high affinity binders are also going to be processed efficiently due to the coevolution of the molecules. And so we recommend just kind of using this as an additional filter, not using this tool separately in a vacuum. And as a recommended alternative, we would say to stick to tools that are trained directly on MHC ligand dilution data like NetMHC pan-EL, because that ligand dilution data inherently takes into account many parts of that pathway. And you can run this tool just as easily as you can the binding tools. So finally, onto the immunogenicity predictions, which again is this part of the pathway, the recognition by a T cell. And this should look familiar by now. We start from the same page. But now instead of selecting binding or processing, we select immunogenicity. And here we only have one context-specific parameter that can be adjusted. And that's related to the positions in the peptide to be masked. And we have a default which will mask the most frequently, the most common anchor residues in a MHC peptide sequence, so 1, 2, and the C terminal. And there are a couple of other options in there as well, including an allele-specific option. When we run this tool, so this tool was trained on a data set of human and mouse peptides where a fraction of those peptides were immunogenic and another fraction was non-immunogenic. And in that study, we simply looked at the enrichment or depletion of all of the amino acids in both of the fractions. And we noted a significant enrichment of tryptophan, phenylalanine, and isoleucine in the immunogenic fraction, and an enrichment of serine, methionine, and lysine in the non-immunogenic fraction. And this tool uses those enrichments to calculate these propensity scores for the likelihood of a peptide being immunogenic. And that score will range from -1 to 1, where lower means more likely non-immunogenic, and higher means more likely immunogenic. So the output of this tool will be a table, again, with 1 row per peptide and allele, and now we have an immunogenicity score column. And again, higher scores mean more likely to be immunogenic. So as with the processing tool, we recommend also not using this tool in a vacuum, but using it as a filter when you require more specificity than is given by the NMHC-PAN EL or BA tools. So what we're showing here on the right is the performance of this tool on a test data set where we have the immunogenicity scores for the epitope fraction on the left and the non-epitope fraction on the right. And we can see there's clearly a difference in the distribution, but you can't really draw a clean line or a line that cleanly separates the two. And when we measure the AUC value, we get between 0.65 and 0.69, depending on the test data set that we use. In comparison to our MHC class 1 binding tools, those AUC values, depending on the allele, are in the range of upwards of 0.95 to 0.98. All right, so those are the 3 main components of the T-cell class 1 tool. And now I'm going to just take a step back and talk a little bit about the T-cell class 1 tool as a whole, and how it's put together. So what we're looking at here are 5 different entrance points, 5 different web pages on our legacy site, on our classic site, where we have tools related to T-cell class 1. And what we realized was that a lot of these tools have a very similar interface. They take in similar inputs. They'll take an antigen sequence, they take alleles, they might take lengths, and we thought it would be good to kind of unify the interface and make all of these tools available on the same page and use the same input parameters instead of having to go to multiple different places. And so that's kind of the motivation behind this T-cell class 1 tool, where we have this one common input where you can put your sequence, you make your allele selections and your length selections, and those parameters will drive the predictions for all of these different predictors that are selectable down below. And in the previous slides, I've gone ahead and made my predictions independently, right? I first made my MHC binding prediction, and then I independently made a processing prediction, and then I made an immunogenicity prediction. And that's great, but we don't have to do that. We can, if we want to make those predictions for the same data set, we can do that all in this one interface. So we can now enter in all of our parameters, our sequence, lengths, and alleles. And we start with our binding predictor, or here is our elution predictor. And if we click on this add another prediction dropdown, we can now add more predictors to be applied to the input data and parameters. And so here we've selected now the processing prediction and the immunogenicity prediction, and we click run. And now instead of running one at a time, all of those predictors will run at the same time, and we'll get an output table that looks something like this. I've trimmed out some of the columns because it gets really wide when you have a lot of methods, but more or less it will look something like this. So now we have 157,000 rows as output for this data set, and we can go ahead and filter and sort to make to kind of get at the data we're most interested in. So for instance, here I filtered by median percentile rank, an immunogenicity score, and a processing total score, and I've gone from 157,000 rows to less than 300 rows to work with downstream. So when we make those predictions, when we make any predictions, we will also have access to a pipeline map, which will show up on the left-hand side of the screen. I think Bjoern is going to show a little bit more about this later. And that will include a few links that I'll also talk about in a couple of slides from now. But those links will allow you to share these results and save them for later use. So all of the predictions that I've shown you so far have been done in the T-cell class 1 prediction tool. And what we've built into this interface now is to be able to take the output of one predictor and send it as an input to the next and kind of build these data pipelines that flow from one tool to the next. And so here I'm just going to give a really quick demo of how we would build a pipeline using this interface. So starting from our T-cell class 1 tool, we already have results, and we know that because we have this little table icon, which means that the results have been loaded. If we want to, after we're done filtering, if we want to now send our results to another tool, for instance, the cluster tool, we would click on this little plus button and go down to the tool that we want to send data to, and then it asks us which column from the table do we want to send. And here I'm selecting the peptide column from the peptide table. Once we click that, we will end up with another box below the T-cell prediction on the left-hand side. So it shows us that we've added epitope cluster analysis to our workflow. And then the data column that we're going to forward to that tool is highlighted in blue. So here is the peptide column that's highlighted. If we scroll down the screen a bit, now we can see that we have the input controls for the cluster tool since we've selected the cluster tool. But what you'll notice is that we do have some parameters we can select, but there's no input box here. So we don't have input data that we're going to give it in this interface because we're going to take the output from the class 1 tool as input. So we click run, and then we'll get our output table from the cluster tool based on the input data, which was the output of the binding tool or the elution tool. And we can continue doing that, building our pipeline until we have something that looks like this, where we've started with T-cell class 1, we do some filtering, we send that output to the clustering tool and do some additional filtering, send it to PepMatch, and then finally to PepX. And then I have a link to this specific pipeline here that you can play around with in your browser and see how it works. Because the best way to understand this is to kind of fiddle with the system and see how the controls work. And then the last thing that I wanted to show you is how to use the sharing functionality. So this is a little movie here. And what I want to show you is that if you go to the pipeline map, and if you've entered an email and a name, you'll see that there. But you can go ahead and click the pipeline link. When you open that in a new tab and paste it in and hit enter, the page should load up with the pipeline in exactly the state that you left it. What's interesting is so you won't see the pipeline name or email, so that you can safely share these links without sharing email addresses. And you'll see that the pipeline loads up with all of the data and parameters attached. We go back and select the next link, which is copy the pipeline configuration, open that in a new tab, and hit enter. The pipeline will also load up, and we'll see again, we don't have an email or pipeline name attached. We also don't have the input data or the output data. But what we do have is the all of the parameters that were selected so that you can take these links and you can share them and you can save them and then you can reuse them. So you can apply the same pipeline workflow to different data sets downstream. So just to quickly recap, the NextGen tool site hosts the complete new T-cell class 1 tool suite, which consists of the MHC binding and elution, the antigen processing tools, the immunogenicity tool. Our recommended methods are going to continue to change as the algorithms are evaluated. We don't have a set of uniform thresholds to select peptide candidates. And we can execute those tools simultaneously. We can filter the results, we can share the results, and we can also combine the different tools to build and share reusable pipelines. Thank you.
Thank you so much, Jay, for that incredibly comprehensive look at the MHC Class 1 tool suite. I'm going to give you a minute or so to take a look at the Q&A because there are quite a few questions that have come through. And to start the section 1 Q&A, I'll also invite Dr. Bjoern Peters, who will be helping Jay with the Q&A. So maybe, Bjoern, I'll let you take the first 1 or 2 questions to give Jay some time.
Certainly. Yeah, thanks a lot, Jay, for the presentation. That was amazing. I will start with the first question from an anonymous attendee. Do the tools support non-canonical or unknown amino acids and PTMs? The short answer is no. We've done actually substantial effort in trying to develop specific tools that deal with specific post-translation modifications. And hopefully they're going to come up in a while. Right now, our recommendation is to put in an X or an alanine or something if you know that you're going to have to do the presentation with it. But overall, unfortunately, currently the tools do not deal with this properly, primarily because there is just too little data out there that that we need to train the kind of machine learning algorithms that we use to deal with these. Another question that came up is from Hassani. How do you suggest handling rare HLA alleles or known alleles, but with little data due to bias in available data? So overall, the approach that we are using here, specifically with the NetMHC pan approaches, is representing MHC alleles as a pseudo-sequence, which is representing the MHC molecule by the residues that are actually in direct contact with the peptides that they're being bound. So even if an allele is rare, if there are sufficiently good neighbors, or if the neighborhood of sequences of likely of similar MHC molecules is well represented, then we have not found any problem. And overall, at this point, the sequence space for human HLA is extremely well covered, and we do not consider it a problem, or we do not observe massive degradation of prediction performance when you go outside of the very well classified alleles. Now, this is different when you go to species like cattle or so, where there's much less data available, and that might be important. But for human and for mice, which is the vast majority of predictions being done on this side, we don't really see the rareness of alleles to be a big problem. Jay.
Yep, thanks. Okay, I picked a few here. All right, so the question came in from an anonymous attendee. Says, can you elaborate on the reference set of peptides that is used to compute percentile rank transform? In particular, the rationale of choosing particular reference set. So this reference set was selected randomly from UniProt, and it's a set of 10,000 peptides derived from proteins across the tree of life. Yeah, so I hope that answers the question.
Okay, I can take the next one. Anonymous attendee, what is the biological rationale for the magic 1% percentile rank threshold? So yeah, it's magic, but also it's based on data. So the way we have been these thresholds have been identified in the past has been essentially taking known and conformed T cell epitopes and then asking what are the binding features that are associated with it. And the first one was this 500 nanomolar threshold that was established, I think, in more than 20 years ago, 30 years probably at this point. That was a threshold that captured 85% of all T cell epitopes bound with 500 nanomolar or better. So this gives you also an idea. This is not like a 100% crystal clear cutoff. The 1% actually matches the 500 nanomolar. So you are in a similar range. Around 90% of all known biologically confirmed T cell epitopes have a binding affinity within the 1% for that allele. And as Jay was showing you, different alleles have different uh, nanomolar thresholds. The 1% one seems to hold better across different alleles. I hope that answers it, Jay.
Yep. So there's a question from Clarissa that says, will I have the same results by using the old tools or the next generation tools for prediction of class 1 or class 2 epitopes? And so the short answer is yes, but it depends on the method that you select. So if we have the same method and version between the between the classic tools and the next generation tools, then the results should be identical between the two. And in many cases, we do have the same methods and versions, but in the next generation tools, we've been dropping some of the older versions of tools. So some of those you will only be able to access on the legacy site or classic site. Sorry, I keep saying legacy. Thanks.
Uh, Thanks, Jay. The next one I have here is from Hassani. Hi, thank you for the great tools. Do you update MHC PAN training set for every release? Can you highlight the difference between the latest release and the one described in the paper? So we do not update the prediction methods on every release. The version numbers that you're seeing on the site should be corresponding to published papers. So we've been sticking with that. Essentially, typically retraining requires quite a bit of manual intervention with the data set or so. So we do that, retrain, and then as a new published version becomes available, it'll eventually end up on the IEDB website. There might be a delay, as mentioned, I think yesterday, because it takes some resources to implement any kind of new update for a tool. I think that's it, Jay.
Yep. And so there's a question from Hassani. It says, since presentation score is the sum of TAP and affinity, can I ignore it and rely on affinity alone? I mean, that's an interesting question. So if you rely on the affinity alone, that's essentially going back to just using the MHC binding tool, and then the processing tool is not adding anything on top of it. So if you want to take into account processing prediction, then you need to use the TAP scores, the processing scores that come out of that tool.
Thanks, Jay. Next question I have is, in literature, I often see HLA AO201 has P2 and P9 as anchor binding position. Have you seen cases where P1 is also possible binding position? So the concept of anchor positions is really a way for humans to communicate easier how different MHC molecules differ And those are the most important parts for binding, but there is always what used to be called secondary anchors. And P1 is a very well established secondary anchor for HLA-A0201. Overall, I wouldn't take these anchor position things too seriously. Again, like they're a way how to quickly like summarize preference between different alleles, but di slight differences in secondary anchors can be very important, and that's what differentiates many of the most commonly used alleles. So we've definitely seen this. Um, I think that was it, Jay.
Yep. So there's a question. Oh, just disappeared on me. Okay, so there's a question from Sumanshu Paul. What is the median percentile and percentile score, and which one is best for taking into consideration as it confuses due to lots of rows in the results table? Right. So went over the percentile rank and how it's calculated in the slides. I know there's confusion. Sometimes we have a percentile column for each individual tool. So every tool that if you select multiple predictors, you're going to get a score or a IC50 value, and each of those is going to have its own associated percentile rank. And then the median percentile rank is simply the median of any of the percentile ranks that are calculated for those from those tools. If you only have one tool selected, the median is going to be the same as the percentile rank for that particular predictor. Bjorn?
Thanks, Jay. So next one is anonymous again. In cross-reactivity scenarios, if you had to pick affinity or presentation, which one would you recommend using? That is, peptides have high affinity to the target allele or only those presented. My understanding, since MetMHC-PAN doesn't consider tissue or gene expression, it would be hard to understand what presentation actually means. So there's multiple parts to this question. Cross-reactivity is always a challenge, and doing clear guidance on how, I mean, cross-reactivity is something obviously the immune system is trying to avoid, specifically cross-reactivity from, say, a viral peptide to the host. And I would probably, if I'm studying this, go more with binding affinity than the presentation score. Now let's clarify though what that means for NetMHC-PAN. So there is these two outputs, which are the BA binding affinity and the EL, the eluded ligand scores that come out of NetMHC-PAN. And the difference is really that the second is trained on eluded ligand data, which essentially adds the processing kind of components to the score. So the kind of preferences that are there due to proteasomal cleavage, tap transport, and all these things are kind of summarized and added on the binding affinity for in the NetMHC-PAN EL score, which is what makes that a preferred score. Um, in the cross-reactivity scenario, something is going wrong. It's like if you're talking about host versus viral peptide, ideally you shouldn't even have T cell population recognizing a viral peptide that cross-reacts with host. So something somewhere is going wrong, and uh, um, so it's a little harder to recommend like how exactly that should be tackled. And this might actually be going too deep, but I hope I answered at least parts of the initial question. Um, Jay, and sorry, every time I click done on this, your stuff is going to move up because
Yeah,
your
I
question is
figured that
jumping
out.
away.
That's okay.
Sorry about that.
Uh, all right. Yeah. So the next question I see is from Mildos Nakalauskate. Sorry for butchering the name. Can this MHC1 predicting tool be used for binding affinity prediction in peptides eluded from murine MHCs? Yes. So I believe there are 6 or 7 species that we have data for that we make available for prediction with NetMHC-PAN EL, so it's been trained on elution data from, there's definitely plenty of mice data in there. So you can just go through the allele selector and you'll find all of the H2 alleles. Bjoern?
Yep, next one I have here is anonymous again. Papers cited for proteasomal cleavage and immunogenicity prediction are from 2013. Are there better methods and data that have been collected in this space and better alternative tools elsewhere? So for proteasomal cleavage and tap transport and all the kind of processing things, there has been this shift. I mean, this was my PhD. I worked on these tools. And overall, training very specific tools for these specific parts of the pathway has been challenging because those data sets are actually really hard to generate, and there's few data sets out there that have like high quality data. In contrast, MHC ligand dilutions, you have hundreds of thousands and millions of peptides that give you like a really, really, really large database that implicitly capture the kind of like processing motifs that are out there. I mentioned that in the last question, and so those have essentially taken over, and we have not been able, and believe me, I tried to come up with better combinations using combinations of explicitly modeled steps in the pathway that then uh, give you a better prediction than the kind of like heuristic summary that you get from something like an EL score. Um, that
All
answers the
right.
question, I hope. Jay?
Yeah, next question. Anonymous attendee, is there a standalone version of the immunogenicity prediction? Yes. So on the next-gen tool site, there's a downloads page. If you go to the downloads page, it's in the immunogenicity prediction is part of the T cell class 1 download. So I would recommend going there. I believe we may have a beta version on the classic site, but I would stick to the next gen version.
Okay, next one's anonymous again. I wonder, given the hype for structures, if there's still a valid argument to search peptides based on sequence similarity and use structure, if at all, as annotation. My line of thinking, most available structures are predictions, so sequence similarity is the closest to reality. Okay, I'm not completely sure I'm following this 100%, but I do want to point out, I mean, what we found, and we've published actually in extensive negative finding papers where we had tried to use structural predictors for improving MHC binding and presentation predictions. And we actually found that we just couldn't get it to improve upon sequence-based comparisons only. Now, I have to point out here that we believe this is really due to the fact that the structure of the peptide presented by the MHC is extremely constrained. The structure of the peptide presented by the MHC is tugged in. There is no movement. There's very, very little movement, if at all, of this peptide. It has nothing to do with the native structure of the that this peptide has in the protein. So, in contrast to antibody epitope recognition, where structure plays a key role for T cells, we just don't see that it adds that much value. We haven't, again, like, so when we say this, these are not opinions formed in our heads, but we are always we try to make the tools better, and then we benchmark them on unseen data sets to see, do we actually get an improvement? And that is our criteria to saying something doesn't work. And doesn't work obviously just means we haven't found a way to get it to work yet. And there's a lot of public tools out there that claim improvements with structure, and and we have not gotten that to run and show a clear improvement as well. And Nina is coming up, which means we are out of time and overtime, and I think we are.
I
So
did,
we're gonna have to answer some of these in typing later.
if that's okay, yeah, I gave it an extra 5 minutes to the Q&A because we do still have quite a few. So I think if it's okay, unless Jay, you want to answer one more burning question.
Yeah, it can wait. Yeah, there's a lot.
So we will type a response, or if any are relevant to the next Q&A, we can answer them in the following Q&A.